Welcome this morning in the neuroanatomy lecture, and today I have to introduce you a field of neuroanatomy, neuroscience, which is not really neuroanatomy, and it is also not really neurohistology, so it's somewhere in between. But there is a topic about this in the list of exam questions, and therefore we have to say something about this. The limbic system and the structure of the hippocampus is to be discussed today, and as always I have to start with some basics. I will define what does it mean, limbic system. I will list you the most important limbic structures. We will talk a bit about the connections of the limbic system. I will tell some afferent pathways. I will tell you some inner connections and some efferent tracts related to it. And at the end, I will point out two areas, the hippocampus, and I will tell you some tracts related to that. And finally, the amygdala will be mentioned. I have to tell you in advance that one could give even series of lectures about this topic. The literature is really huge. What I would like to tell you is maybe to impress you, to introduce you a bit this part of neuroscience, and those slides which are really exam related points I will always focus on, and I will tell you which slide is absolutely required in the exam, but some remarks are not as important as others. So this is important, of course. What is the limbic system and how to define it? So the limbic system, the term comes from the Latin word limbus. The limbus is the border of something, the edge of something, and when people started to study the brain histologically, they prepared histological sections from all over the brain tissue, and it turned out that if the tissue sample was taken from the borders of the cortical surface next to its end, its rim, its edge, people couldn't find the typical 6 layers of the cortex, what you are already familiar with. But it turned out that this is thinner and it has less layers. Later, when people started to study the development of the brain, and they compared the brains of different animals at the various levels of the evolution, it turned out that those brains where we have more ancient parts in a higher proportion, so phylogenetically older parts of the brain, will have this simple structure, and it was summarized under the term allocortex, which means an more ancient part of the brain. And people started to use the term limbic areas because they were at the border of the cortex. Now, then science developed, and then it turned out that these limbic areas have really strong connections with other brain areas which are not defined or not classified as cortical centers. Therefore, this term changed its meaning, so we still use the term limbic system, but now we also include several subcortical centers also into the limbic system because of functional connections, as you will see later. And if I want to make it very simple, and I want to summarize very briefly the function, the significance of the limbic system, is that we have the memory formation and the emotional life, so the affective stimuli that we feel, that we can link our feelings with the events, that is related to the limbic system. So the limbic system is a system of functionally interrelated brain areas involved in memory formation and in affective emotional life. This is how I could define the limbic system, but I have to state that there is no real nice definition for that. So it's an anatomy lecture, so I have to tell you the anatomy of the limbic system, and this is an option to review the brain, because there are only a few parts of the brain which cannot be considered as limbic structures, as limbic related structures. So many brain areas can be listed as a limbic structure nowadays. But the main part of the limbic system is the so-called limbic lobe. Also, if you can remember my short introductory talk about the brain macroscopy several weeks ago, I mentioned that the parahippocampal gyrus and the cingulate gyrus together are considered as limbic lobes most of the time. So in this slide, I highlighted the limbic structures. It's indicated in yellow already, but some names I also indicated. The gray boxes will refer to gray matter, and you will see some white boxes coming up. They refer to the tracts, which also belong to the limbic system, because these centers, as I mentioned, are interconnected with each other. So the next area, what I would like to mention, is a very tiny gray matter that covers the surface of the corpus callosum. You know, the corpus callosum is part of the hemispheres. It connects the two hemispheres with each other, and the surface of it is covered by a tiny cortical layer called indusium griseum. It's also limbic structure. The septum pellucidum that separates the two lateral ventricles, anterior horns, and central parts in the midline is also limbic structure. And now we already leave the original definition, because as I told you, one started to use the term because it has been recognized that these centers are situated at the borders of the cortex. But later on, it turned out that the anterior nucleus of the thalamus is closely related to the limbic system. It has connections with the cingulate gyrus, and therefore the anterior thalamic nucleus is also involved. It is an important center in the so-called circuit of Papez. I will come back to this point in a later slide. Then also, if we go down inferior to the genu of the corpus callosum, and in front of the lamina terminalis of the third ventricle, there is a tiny gyrus that is called paraterminal gyrus, and this is the so-called subcallosal area here. In front of it, this is also part of the limbic system. Also, a gray matter area is here next to the parahippocampal gyrus, which is the uncus of the parahippocampal gyrus. You also know already the olfactory bulb and the olfactory tract, the olfactory trigone, which are found in the inferior surface of the frontal lobe. They belong also to the limbic system. I will come back to this point. Also, I will come back to the amygdala, which is an important center connected with several other brain areas, and it has to do with emotions as well. The mammillary body is, again, a hypothalamic structure, diencephalic structure that is part of the limbic system. I will discuss with you the connections of this. Parahippocampal gyrus, I mentioned already, that belongs to the limbic lobe, and next to it we have the hippocampus. I will talk about it a bit later, detail. And then some connections I also highlighted. The amygdala is connected with the hypothalamus, and some fibers will also connect the hippocampus with the mammillary body, and from these tracts some nerve fibers will exit, and they will contribute to the formation of the stria medullaris of the thalamus. I just mentioned the fornix, and the parts of the fornix you are already familiar with, because it belongs to the walls of the ventricles, the medial wall of the central part. If you can recall that in the lateral ventricle was mentioned, that's the crus, and then downwards the column of the fornix contributes also to the anterior wall of the third ventricle, so this is also already familiar to you. Mammillothalamic tract, I will come back to this point. That connects the mammillary body with the anterior thalamic nucleus. It's also an important limbic pathway. It has to do with memory. I will come back to this. The mammillotegmental tract connects the mammillary body with the tegmentum of the midbrain. Stria terminalis is the connection between the amygdala and the hypothalamus. I will come back to this also. And you are already familiar with the anterior commissure. That is a connection between the right and left hemispheres. It's a commissural pathway. It connects temporal areas, and also olfactory information is exchanged between the right and left sides of the brain. And importantly, here, this dotted curve or line would like to represent the brainstem, and it's not highlighted with yellow color in this drawing, but inside the brainstem we also have some nuclei which could be listed as limbic structures like interpeduncular, tegmental, and Raphi nuclei. The Raphi nuclei you are already familiar with in earlier lectures in the brainstem, in the reticular formation. You could hear about these centers. So each of which has its own significance, and I have only limited time, so I cannot go through all these systematically, but I try to summarize the most important things. So let's go on. After this short listing of the most important limbic structures, I would like to talk about some tracts. And on one hand, I have to introduce something that is not really coming up in other lectures. You have a couple of lectures about sensory systems, but the olfactory system is a little bit neglected, and the vision and the hearing and the vestibular issues are discussed in detail, but somewhat neglected is the olfactory system. And I would like to talk a bit about this because the course director, when I got this lecture a couple of years ago, asked me to introduce a bit also the olfactory system, and because it is a strong input into the limbic system, this is a good occasion to use this opportunity. So olfactory mucosa, that is already something you are familiar with. It's in the upper part of the nasal cavity where you have olfactory epithelium. And in this olfactory epithelium, we have olfactory epithelial cells. They are the so-called primary sensory cells. I gave you a lecture in the early part of the semester about receptors, and I took this example at that time, that here the olfactory information is picked up, and from these cells, the cilia will pick up the signal, and their own axon will carry this information through the cribriform plate into the olfactory bulb, where then the information is signalized further to the second order neuron of the system. So olfactory mucosa, olfactory fibers, and we arrive in the olfactory bulb. This scheme shows you an overview of the olfactory bulb. Don't waste your time too long with the connectivity of these cells. If you are interested in that, you can see that we have different kinds of neurons here, periglomedular cells, mitral cells, and granular cells, and they are quite complex in a quite complex synaptic connectivity. I don't want to go into detail. It's also not exam related, just to demonstrate that there is also a complex system here. And then after a couple of synapses, the primary olfactory information is perceived by these cells, and they will start the olfactory tract. Now, the olfactory tract has also nuclei, and there is a so-called anterior olfactory nucleus from where the information is also crossing the midline, and it goes to the opposite side. So right and left connections we have, and in us humans, this function is somewhat subordinate because we do not really have that ability, or at least that it is not very strong in humans, that we can determine the direction of the olfactory information. But in animals, it's very obvious that they exactly know from what direction the olfactory signal is coming. So you can see dogs that they can follow the traces of other animals or people, because they feel simply the concentration of the olfactory signals and this right and left connection, and allows them to compare the right and left nasal cavity and how much olfactory information comes in, and based on this, the animals can determine from what direction the smell is coming, so they can find the other animal, for instance, if they want to catch, for instance, predator animal, for instance. Okay, then we arrive to the olfactory bulb, where we have the second neuron, as I mentioned, and then from the olfactory bulb, we arrive to the olfactory tract. And then from the olfactory tract, we go on And we come to this olfactory trigone here. And from the olfactory trigone, we have multiple directions. One direction is that we go to the lateral side, and that's the lateral olfactory stria here, which then goes straight into the olfactory cortex, which is called preform or preform area. And there you have small gyruses. You won't be able to demonstrate these gyruses on the brain because they are very small and well hidden, covered by the temporal lobe, but they comprise the primary olfactory cortex. It's made up by the ambient and the semilunar gyruses, from which then the signal goes on into the amygdaloid body, into the amygdala. I will talk about the amygdala a bit later also. And this is one direction. And please note that I didn't mention the thalamus. And you heard about the thalamus, that those information sources which reach the cortex, which go into our consciousness, will go through the thalamus. And the only exception is the olfactory pathway, because without having passed the thalamus, the information spreads into the cortex. And from here into the parahippocampal gyrus, the signal may continue, and then this is an important center of the limbic system, so it can influence our feelings or emotions. And in us humans, we think that we didn't know for a longer period of time that we are quite strongly influenced by our olfactory system, but mostly in humans this stays at the unconscious level. But if you find somebody attractive, somebody you identify as a potential partner in your future, that depends also, at least in part, on olfactory information, not only on visual signals. And this has to do with our feelings. And if you can make a strong partnership, strong emotions you can produce in your mind in relationship with the person, is depending also on olfactory information. So that's why the olfactory input is quite important for the bonding and for the partnerships in order to make up relationships. And also there is another direction to the medial side. That's the medial olfactory stria. It goes to the subcallosal area and the arterioperforated substance here. And this has to do with nutrition and feeding, because the olfactory information will also decide whether or not we eat something or not. So if you open the fridge, you find something there which you cannot remember for how long time is it already there. You open the box and you smell it. And if it smells like hell, you won't eat it. So the nutrition is also pretty much influenced by olfactory signals, and they may cause also quite strong emotions if something is really uncomfortably smelling. And also the anterior perforated substance has other functions, but I don't want to go into more details now. And I mentioned that the anterior olfactory nucleus situated right behind the olfactory trigone and partially inside that. You have it has also back and forth connection, so it goes back also to the olfactory bulb, so it has a kind of feedback function to the olfactory system, and can influence the signal transmission also. So these three directions exist if it's about the olfactory pathways. And then I would like to continue with this subcallosal area story. I couldn't finish this in the last slide because it was already busy enough, and this has to do also with nutrition, with appetite, and with preparation for feeding. Since if the signal comes from the subcallosal area, either through the indusium grisellum, I mentioned this in my earlier slide, you have a pathway that is called longitudinal stria that wraps around the corpus callosum and goes into the amygdaloid areas. On the other hand, through the stria medullaris of the thalamus, the information also goes into the habenular nuclei, and later on into the interpeduncular nucleus that is in the tegmentum of the midbrain. And after these centers, there is a pathway arising, which partially comes also from the hypothalamus, and that is the dorsal longitudinal fasciculus. that goes down into the autonomic centers, controlling the saliva production and also the gastric functions, like the dorsal vagal nucleus and the superior and inferior salivatory nuclei are controlled by that. So if I feel the smell of my favorite food, my favorite cookies, for instance, then I will start to produce immediately saliva, and my stomach is preparing for the feeding and starts to produce hydrochloric acid and also other substances required for digestion. So this connection will influence the appetite, will influence the nutrition and the food uptake, and the whole thing starts with the olfactory information. Okay, so this is just one highlight of the olfactory and the food intake significance of the limbic system. One could give more details, but I think it's enough. I will come back to these tracts once again a bit later, but now let's go on to the other main afferent source of the limbic system, because the limbic information is also coming from the periphery, not only from olfactory impulses, but also from the spinothalamic system. And you most probably remember that we defined the spinothalamic system as a system for protopathic sensibility, for crude touch, for pain temperature. And this crude touch point is that what I would like to discuss a bit more detailed now. So the crude touch, and that our skin is coming into a certain activity by getting mechanical stimuli, is important for the brain and also for our emotions, because if somebody touches our skin, that evokes really strong emotions in us. It might be positive or it might be negative. So if you have a partner and you are coming together and you had a busy day, you are tired, or you're just upset, and your friend or your partner hugs you around or just touches your back, that will cause really nice, positive, strong emotions, and you get relaxed and you feel yourself better in a minute. Or on the other way around, if somebody is touching you and you don't want that, and it's a person who you don't even know, that evokes anger and really strong aggressive emotions in you, which is also very common. So this shows the connection between the spinothalamic system and the limbic system. And the spinothalamic system is much more complex that we discuss usually, because we always talk about the pathway that comes from the dorsal horn of the spinal cord and goes to the VPL of the thalamus and then to the cortex. But besides that, we also have fibers which reach the reticular formation. It's synapses in many nuclei. Don't waste your time with the names of these nuclei. It's enough if you know it's the reticular formation. And then from the reticular formation, after several synapses, the information goes on and reaches other nuclei of the thalamus also. For instance, the centromedian nucleus, but also it reaches through a different pathway the so-called medial forebrain bundle, the amygdala, and the septal nuclei, and they will belong to the limbic system already. And this is how the crude touch and touching your skin will cause strong emotions in you. And this also has to do with sexual behavior and also sexual arousal, that the surface of the skin, if you touch the surface of the skin, causes very, very, very strong emotions, and emotionality is related also to sexual behavior. All right. Then after this input of the limbic system, let's talk a bit about the inner connections and about efferents. And the next slide is, I think, the most important slide in this lecture. And if you learn anything from this lecture, this slide is that what my colleagues will ask you in the exam, and I also do this. So inside the limbic system, there are many centers, as you could see in my second slide. It was yellow, you know, the half brain was yellow. But this neural circuit, which was identified already many years ago, by a person who was called Papes, but most of the times people pronounce it as Papez neural circuit, but this person called himself Papes. But if you say Papez neural circuit, everyone understands what you want to say. So this neural circuit is in itself returning neural pathway. So it starts somewhere and comes back to the same point. So one could say, what kind of sense does it make if something is like a circle, it returns? The sense or the rationale of this is that if a signal enters this system, and if you want to make this as a memory, and if you want to remember something, and if you repeat this information multiple times, and if you meet this information, or it is loaded by stronger by emotions, then the information will run much more circles in this system, and then there is an opportunity to rearrange the synapses through the plasticity. This is what I mentioned a couple of weeks ago about the synapses, that they can be re-established, they can be rearranged, and this helps the memory formation. So the more times the information goes in this neural circuit, the more likely is that you will remember it for a longer period of time. But if this neural circuit is interrupted, that will, of course, interfere with the memory formation. So that's why you have to know this circle. You could start anywhere because it's a circle, but I think it makes sense to start with the hippocampus. From the hippocampus, the mammillary body comes. The connection between them is the fornix. And then from the mammillary body, the mammillothalamic tract goes into the anterior nucleus of the thalamus. From there, radiation, the thalamo-singular tract goes into the cingulate gyrus. And within the cingulate gyrus, in the white matter below the cortical surface, is a pathway called cingulum running. And this cingulum wraps around the splenium of the corpus callosum and reaches the parahippocampal gyrus. And from there, there is a perforant path which reaches the hippocampus, and we came back into the original area. Okay, so this is the neural circuit of Papez, and this is which has to come into your mind if it's about limbic system, because this is required for the memory formation, and this is the most important pathway or system of pathways. For a better mark, if you would like to make a really good impression in the exam, you may remember some other tracts. I mentioned already the stria medullaris of the thalamus. This connects the septal nuclei and the olfactory system with the habenular nuclei, and it runs on the thalamus. You may remember that the roof of the third ventricle, the tela choroidea of the third ventricle, was attached to the stria medullaris of the thalamus, which is a limbic pathway. And then the stria terminalis is another thing what I mentioned. This is a connection between the amygdala and the hypothalamus. The hypothalamus has centers for autonomic functions, and if I see something that is potentially dangerous for me, that evokes strong emotions in me. It's a life-threatening situation, for instance. There is a very bad, dangerous situation occurring in the traffic when you are driving your car and you have to avoid a car crash or something. Then the brain is immediately alarming the autonomic nervous system. The heart rate increases, the breathing gets accelerated. This is done by the stria terminalis, because from the amygdala, the hypothalamic centers will get information through the stria terminalis how to adapt to the environmental challenges. Then there is another tract which is not indicated in this scheme. From the habenular nuclei, there is a tract going down into the interpeduncular nucleus. It was indicated in that scheme I showed you earlier with the saliva production and the nutrition. This is called habenulotegmental tract. Because of the curve of the tract, it's also called fasciculus retroflexus. If you read this, it's also found in books. But once again, these are the less significant pathways. The Papez neural circuit you must know. If you can list these, we are even more happy, but they are less significant. And I also mentioned that we have efferent connections, and one could give several lectures only about the efferent connections, and I only have a few minutes for that, so I won't be able to discuss this systematically. Until now, this slide shows nothing else than the circle of papes or puppets, as I mentioned. So once again, hippocampus, fornix, mammillary body, anterior thalamic nucleus, cingulate gyrus, hippocampus. The green arrows refer to the tracts I showed you in the earlier slide of mine. So this is where the information runs around. So if you would like to call somebody and the cell phone number is not stored in your cell phone, and somebody tells you the numbers, then until you are typing these numbers with the keyboard into your cell phone, you can maybe remember. But if you don't repeat it, you don't review this several times, you won't remember the cell phone number after half an hour for sure. But if you call this person every time and you don't use the saved number, but you type it again and again, you will remember after a longer period of time those numbers, because it will go from this working short-term memory into the long-term memory. And we don't exactly know where the long-term memory is stored in the brain. Most likely it's diffusely stored everywhere. At least the association cortical areas are involved in that. And diffuse cortical damage may lead also to long-term memory deficit. And this is the place from where you can recall also the memories, either through the cingulate gyrus or through the hippocampus. Therefore, please note that the hippocampus is the access, an important center for the memory access. So if we lose the hippocampi on both sides of the brain, we are unable to memorize things, and we cannot store new memory traces anymore. An amnesia occurs, which means that after the loss of the hippocampi, we cannot remember new information, and we cannot build up new memory traces. Therefore, the hippocampus is a critical center for memory formation, and it is the access to the memory. Also, we go from here into the amygdala, which has emotional memories also. And as I mentioned, from here, the stria terminalis carries the signals into the hypothalamus. The fornix also sends fibers to the hypothalamus, which controls the endocrine functions and the feeding. And if you are in a stressful situation, you know that you lose your appetite. If you are after a stressful period, but it's over and the exam period has ended and you relax a bit, your appetite returns, and then your personality, your behavior will change, because the stria terminalis has to do with the center located in the basal forebrain, the nucleus accumbens. This is an area where the head of the caudate nucleus and the putamen are kind of coming together. And from here, this is the basal forebrain. From here, then we go into the prefrontal cortex, which has the personality, the behavior, and also the emotional drive, in a way that if you have the initiations, if you can start to perform something, depends also on the prefrontal cortex and how you behave in a certain situation. And this is also controlled indirectly by the limbic system and by the amygdala. So if you have strong stress, if you have strong pain, if you are really worried about something, you are worried because of a relative who is sick, then you are more angry, you are a little bit more aggressive, or sometimes even very aggressive. So this is related to the connection with the limbic system, and it interferes also with our behavior. If we have pain, or if the patient has pain, might be aggressive. So if a patient is transported to the hospital because of any disease, first you reduce his or her pain. Of course, you don't want that he or she suffers, and you also protect yourself, because the aggression of the patient is also related to the pain, and that comes from the amygdala, and the prefrontal cortex will determine the personality. And in a short period of time, because of strong emotions, the behavior may change. Okay, and then after this, I would like to talk about some functions and some limbic structures a bit more detailed. And the talk is about hippocampus also, and I have to tell you something about the hippocampus and hippocampal formation. So one thing is, what people usually don't know, that the hippocampal formation is not a synonym for the hippocampus. The hippocampal formation is more than the hippocampus proper, because if you take the knife and if you remove the temporal lobe from the rest of the brain and you open the lateral ventricle in a superior view, you will see that we have the hippocampus here in pink. Then you have here next to it the dentate gyrus. And after this, you have the parahippocampal gyrus, which contains areas called the subiculum and then the entorhinal cortex. And these together will form the hippocampal formation, and the hippocampus is just this part. So the hippocampal formation is more than the hippocampus because it involves the dentate gyrus, hippocampus per se, the subiculum, and the entorhinal cortex. Also, there is an alternative name for the hippocampus. It is also called cornu ammonis, or Ammon's horn. And this is something that I have to explain you. And also, it makes sense to mention why do we call the hippocampus hippocampus. And this is the preparation of Professor Sheresh, who is a professor on this university. He was the head of the laboratory for electron microscopy for many years. And he's a quite acknowledged hippocampus researcher. And this is a drawing of him, or a preparation of him from his book. So if you remove the hippocampus and you cut it out from the surrounding tissues, it looks like a seahorse. And therefore, because the seahorse is called hippocampus, we also call this brain area hippocampus. So this is a specific name for the brain area. And as I mentioned, it's also called cornu ammonis. This has to be explained also, since if I take the knife and if I cut a frontal section of the brain, one can see that the hippocampus has this curvy structure. And after this maneuver that I cut it in a frontal plane, it shows some similarities with the horn of a ram. And in the Egyptian mythology, the ram is in the sculpture of god Ammon, who is an important god in the Egyptian mythology, and therefore the horn of Ammon, or cornu ammonis, is the Ammon's horn. Is also an anatomical term that we many times can find in the literature. If we zoom in into the frontal section, again, one can see the dentate gyrus, the hippocampus, the subiculum, and the entorhinal cortex in the parahippocampal gyrus, as I mentioned. So this is in total, altogether, again, the hippocampal formation. Okay, if we go on, one slide I have about the synaptic connections of the hippocampus. This is again not the most important slide of the lecture. So if you don't go into details here, nothing happens. But if you want to get a good mark in the exam, my colleagues may ask this also. So the hippocampus is cut in a frontal view now. And the areas are defined inside that. They are CA1, CA2, CA3, and some areas, some people also define a CA4 area. CA stands for cornu ammonis, what I mentioned earlier. And it has pyramidal cells. These areas have pyramidal cells, which have axons, and they form the efferent pathway of the hippocampus. But how does the information arrive here? From the parahippocampal gyrus, the so-called perforant pathway comes, and it synapses with the granular cells of the dentate gyrus. And these granular cells will synapse mainly with the apical dendrites of the pyramidal cells here. And from the pyramidal cells, the efferent pathway arises, which finally contributes to the fornix. And if I cut this area and if I zoom in, one can find here the layers. The alveolus hippocampi is the white matter where the fibers will go. And then we have the stratum oriens, which is a layer. Then we have the pyramidal cell bodies layer, pyramidal layer. Then we have the radiatum, stratum radiatum, which contains the dendrite tree. You have a lacunosum, stratum lacunosum layer. And also we have on the surface a molecular layer. So these are the layers of the hippocampal cortex, and you can see that we cannot distinguish 6 layers. So that's why it's a holocortical area. This is what I mentioned in my introduction. In this slide, I list some examples for the hippocampal connections. At least some examples, please learn for the exam. The main connectivity is related to the neural circuit of Papez. This is what I already mentioned, so I don't want to tell you again. So one can see here the cingulum, which arrives to the parahippocampal gyrus, and from the parahippocampal gyrus we go into the hippocampus. This is the perforant path, which I indicated in the last slide also. And then from here, we go through the fornix into the mammillary body. This is the main direction, but also other hypothalamic nuclei get informed about the limbic issues through the fornix. Also, some fibers go backwards into the through the cingulum, but I don't think so that it makes sense to talk longer about it. Most important connections are from the parahippocampal gyrus, and they go to the mammillary body. These are the most important ones, and some other examples are also mentioned here. Okay, and when I was talking about the functions of the limbic system, I mentioned the memory, and you will learn about the memory a lot in the physiology, but just to make you a little bit more familiar with this, there is an explicit type of memory, and this is where the hippocampus has a key significance, because this is involved in learning, and this is the declarative type of memory that you can remember events, that you can remember facts, concepts, and this is what you need for learning. So if you sit down to study something, this is what you definitely use to remember semantic, or to produce semantic or episodic memory traces. This is where the hippocampus is absolutely required, but the hippocampus is not properly, it is not responsible for the storage. It is a kind of access to the memory, so it will be connected, as I mentioned earlier, with the other areas, which will store in part and will contain the memory traces for a longer period of time, and from there you can retrieve the memories. However, if the connection between the hippocampus and the parahippocampal gyrus is interrupted, then you don't have the access to the memory. And Alzheimer's disease is a neurodegenerative disease. You most likely heard about it. It affects several parts of the brain, but many times it starts with or at least it is related to the damage of the hippocampal connections, and therefore the patient has a memory deficit, which gets more and more serious. The implicit memory is not that I would like to talk about today, because you heard about the motoric skills, and the motor coordination is discussed in a later lecture also, but basal ganglia and cerebellar centers are involved in the practiced skills to remember how to do something. This is also another part of memory, but I don't talk about it. But also there is an emotional type of memory, which is related to the amygdala, and this is the final point I would like to introduce a bit, of course, without going into details. The amygdala or amygdaloid body is situated in the temporal lobe. If I indicate here the lateral ventricle at the tip of the temporal horn, there is a cap-like shape of gray matter found there, which has a somewhat similar shape with an almond. Therefore, it's called amygdala or amygdaloid body. And in this frontal section, you can see this gray area here. This corresponds to the amygdaloid body. It has more than 30 subdivisions. Of course, you don't have to know that. I mentioned here some subnuclei with a couple of functions. Again, you don't have to waste too much time with that, but the corticomedial and the basolateral nuclei I pointed out with emotional functions and some connections. If you would like to memorize these names, you may get a better mark in the exam, but this is not absolutely required for passing grade. The amygdala controls our emotions, and this has to do with psychiatry, neuropsychiatry also, because just an example, if something happens in your life which is related to very strong emotions, it evokes very strong emotions in you, you will remember that for much longer period of time. If I ask you what was your breakfast or your dinner 2 weeks ago on Thursday, you won't remember, except if there was something exceptional on that day. You had, for instance, your birthday accidentally on that day, then you will remember what food you made with your friends or with your relatives when you celebrated. So you connected the memory with emotions, and you will then remember for a longer period of time. Also, if you get a topic in the exam, after the exam period, you still can remember that particular topic, what you answered in the exam, but all the other topics, what you didn't pull as a card in the exam, you already forgot, and you have to review it. This is again an example for that the stronger the memory is, the stronger emotions are required linked to that. The exam is a strong emotional situation. You will remember that for a longer period of time. And the amygdala is involved in that. And also you have the amygdala, which is involved in freezing and freezing related symptoms. So if you are in a very stressful, life-threatening situation, then you react with freezing, with increased heart rate, respiration, and endocrine responses. This comes also from the amygdala. And if you influence the function of the amygdala in experiments, then correspondingly the emotional functions will lead to autonomic response and feeding behaviors as well. And if we destroy the amygdala, then this is also influenced. The so-called Cleaver-Bussy syndrome occurs, which is not very common, but it means the loss of fare, this so-called hyperorality, which means that these animals in experiments, or humans sometimes, but luckily not very commonly, put all possible objects into the mouth, which one wouldn't eat. And also the certain kind of hypersexual behavior is also characteristic for these experimental animals. So people this way could identify the significance of the amygdaloid area and the neighboring brain regions. So this is what I could highlight in this very short period of time. I'm sorry for speaking 2 minutes longer. I also put emphasis on those slides, but I really want to hear back in the exam the Papez neurocircuit and the connections and the list of limbic structures, and that you have here memories and also the formation of memories and emotions is the system related to. All right, thank you for your attention. If you have any questions, just let me know.